Okay, this video is complement and pathogens in ALS and a brief summary of what the video is about. Okay, first we're going to look at which microbes are associated with ALS. Then we're going to look at what the literature is telling us about complement activation in ALS. Then we'll be looking at what is complement. Okay, once we've looked at complement, we're going to try and piece together where those microbes that we looked at associated with ALS fit into complement. And the end goal is to figure out how complement is being crippled by these pathogens. Okay, the first research paper we're going to look at is by Dr. Garth Nicolson. And if we look at this particular research paper, Chronic Bacterial and Viral Infections in Neurodegenerative and Neurobehavioral Diseases, pathogens associated with ALS from the Nicolson paper are Chlamydia pneumonia, Borrelia burgdorferi. There's a bunch of mycoplasma infections. There's the herpesvirus 1 and 6. And there's also the evidence of the retroviruses in half the ALS patients compared to only 7% of the controls. So now we have endogenous retroviruses that are actually in the human genome. So Dr. Nath has got a study where he linked this retrovirus, it's called HERV-K, to ALS, and that this is one of the viruses that they're looking at as well, one that's actually in the human genome. The next study is evidence for fungal infection in the cerebrospinal fluid and brain tissue from patients with ALS. And this one here is quite a striking study, because what you find with this one here is when they tested the brains, they found Candida in all 3 of the brains. They also found a few other species. So the species are the Candida, and then there's this Trichoderma viride, and Cryptococcus magnus is the 3rd species that they found quite prevalent in ALS patients. So if we look further at this study, there's one part of the study that the word's quite strong. In our opinion, the most striking finding was that C. albicans appeared in all 3 patients. This yeast was also present in the cerebrospinal fluid, several ALS patients, and it acknowledges that C. albicans can be highly pathogenic. The other thing that you find in this particular quote is that they also found the Trichoderma viride in all the samples. The last one, the Cryptomagnus, was only in 4 of the patients. The next study is Leaky intestine and impaired microbiome in an ALS mouse model, and this is perhaps one of the most profound, more recent studies out. It was published in 2015. In the ALS mouse model, generally the neurological symptoms start at 3 months. What this study did was they looked at the mice at 2 months. What they found at 2 months was reduced tight junction protein ZO-1 and reduced protein E cadherin. Together, these 2 proteins give leaky gut, and leaky gut increases large molecules past the intestinal wall. The next super important finding of this study was abnormal Panth cells. With abnormal Panth cells, you get a decrease in this polypeptide intestinal antibiotic called defensin alpha 5, and you also get a reduction in lysine 1. The lysine 1 damages the bacterial cell walls. Together, these 2 reductions reduce the body's defense of pathogenic gut microbes. So then another thing that's found is an intestinal microbiome shift because of these changes. So you get a reduction in your butyrate-producing bacteria, and these guys feed the colonic EC cells, which produce your serotonin. You also have a reduced E. coli and Firmicutes. Okay, so basically you've got what's known as a dysbiosis, and the other result of this is you have inflammation in the lining of the gut. Another thing that it's got here is a cytokine response of IL-17, and the other thing that this study identified was a dysfunction in the autophagy, or at least a potential dysfunction in the autophagy, which is a huge problem in ALS. The next thing we're going to look at is complement activation in ALS. And the first study we're going to look at is about factor C5a in ALS. And when we look at this particular study, what we find is there's an increase in a complement component C5a There's also an increase in the C3b. There's an upregulation of the C5AR in the lumbar spinal cord. There is also an increase in the C5AR positive astrocytes, and there's also an increase in the C1BQ mRNA found in this particular study. Okay, this next study looking at complement activation was looking at them in the motor end plates for ALS. And what this one found was an increase in the C3. It also found some C1Q, which is activation and complement. It found MAC. It found a couple of regulators in complement, the CD55 and the CD59. And it found C3 mRNA. Okay, so this next study looked at a biomarker 4411, and that biomarker tested for a fragment of factor H, and it found increased factor H in ALS. This next study measured for complement component CD4 and found an increase in the cerebrospinal fluid in ALS patients. Major histocompatibility complex class 1 molecules help to protect against viruses, and what this study found was that they were reduced, and it also found that increase in them helped to protect the motor neurons. ALS TDI is a research facility in the United States, and they have got lots of mouse studies that they've done, and the following graph is from one of their mouse studies. And what you see in this particular graph is you see at 60 days there's a huge increase in the C4b in the spinal cord, in the muscle, and in the brain. And you can see that it continues to increase quite rapidly through the spinal cord and in the muscle there. This next graph is also from ALS TDI, and what you can see with this one is something called galactin-3 is way out of control in the spinal cord and in the muscles in the ALS mouse model. This next paper is suggesting targeting an angiogenin for ALS, as it is the only functional mutation found in ALS, and it results in an angiogenin deficiency. Angiogens are antimicrobial proteins that are also made by the Panth cells, the same cells that made the defensin alpha-5. Okay, these also have properties to protect against pathogenic bacteria and fungi found in the gut. So what we've looked at so far are some of the pathogenic microbes associated with ALS. Then we've looked at some of the studies that are showing complement activation in ALS. And now we need to look at what is complement. So complement is innate immunity. And what complement has is it's got 3 different pathways for initiation. So there's the classical pathway, the Lethean pathway, and the alternative pathway. There's also a 4th pathway called the bypass pathway that's not on this slide. Okay, so they all have different ways for initiation. They converge into the center, which is known as the amplification. So there's huge amplification after the initiation, and then they all have their pathways for termination. To appreciate the implications of how multiple pathogens with aggregating complement interference cripple innate immunity, we need to look at how the system is designed to work. So with the classical pathway, the various proteins were named in order of discovery, and that is different than the order they're activated. And then with the naming system, the proteins are cleaved and the 2 parts are named after the parent protein. So C4 splits into C4a and C4b, and then when they split again, you have more letters added on. So the protein order in the classical pathway, which in the initiation is C1, C4, and C2. And then for the amplification, you get C3, and the C3b, which is split off of the C3, is also fed back into the amplification loop. And then C5 to C9 go in order to make the MAC complex, and that bores holes into the pathogens. In the initiation phase, the different complement components bind together and they form these other complexes that then go on to set off the amplification loop. So you have this alternative pathway that's activating on surfaces, the mannose binding Lethean pathway, which is also the Lethean pathway, makes these microbial carbohydrates, and the classic pathway makes these immune complexes. So these complexes feed into C3 convertase, and out of this C3 convertase you get a huge amount of C3b being made, roughly 10 billion in 15 minutes. Now the C3b then goes around and it deposits itself onto different cell surfaces, both pathogenic cell surfaces and host cell surfaces. The terminating phase is where these cells marked with the C3b are eaten, or they have holes blasted into them, and the other thing that's happening is more cells are recruited. The C3b does not discriminate to what it attaches itself to, whether it be your host cells or pathogenic cells. So then what you have is you have different systems built in for control. So factor H is one of these control proteins that will attach to C3b and protect the host cell. This picture is a far more detailed representation of complement. And if you look at it close, you can see the three pathways: the lithium pathway, the classical pathway, and the alternate pathway. You can see the complexes of initiation that are formed. You can see the amplification loop, and you can see that there's just a lot more different controls, different identifiers on the cells for this particular diagram. You can read up on this from the paper it's from, Complement Activation, Regulation, and Molecular Basis for Complement-Related Diseases. This next diagram represents a healthy host cell. And if you look at this one here, you can see the factor H around the outside of it, and the factor H is protecting the cell from destruction. Here you see a representation for factor H. And what you can see, the one side is for complement regulation, the other side is for attachment to the surfaces of host cells. You can see in the figure B that it's got the one side for regulation in the plasma, the other side for the regulation on the surfaces. And you can see the picture where it is protecting a host cell by attaching itself to the C3b. Overall, this is a very simplified look at complement. So now what we're going to look at is pathogens have all of these evasion strategies to get around complement. So we're going to have a look at what are the evasion strategies of these microbes that are associated with ALS. Earlier, we saw a study that found candida in the brain and spine of ALS patients. And so we're going to look at candida first. And what we find with candida, there is something called PRA1, which binds to factor H and factor H-like protein 1 for immune evasion. This 2013 paper is an exceptional summary of the research on candida. And they also have this really great drawing of candida. Factor H-like protein 1, C4BP, and plasmagen are all captured by candida, allowing candida to control and evade complement attack. There are 4 factor H binding surface proteins, and they're called GPM1, PRA1, HGTP1, GPD2. By capturing the C4BP, the classical complement activation pathway is inhibited. The epithelial lining is usually a sufficient defense to candida. But remember, the first measured ALS symptom is a leaky gut. So this diagram shows epithelial invasion as well as complement. Candida also makes this protein related to something called human vertebrate alpha beta 3 integrin receptor. And this protein acquires a terminal inhibitor called vitronectin. So it is turning off complement attack by acquiring this thing. It also secretes aspartyl protease, and that degrades and inactivates C3b, C4b, and C5. Remember, C4b is from initiation, C3b is from amplification, and it also attaches to things and says kill me, but it's being dissolved here, and C5 is the beginning of the complement attack. Candida interacts to inhibit all of the pathways of complement. If you recall this graph, it shows a huge increase in galactin-3 in the spinal column of ALS patients. This particular study shows that galactin-3 kills candida. We also have that earlier study that showed candida in the brain and the spinal columns of ALS patients. Because vitamin D levels are linked to susceptibility to infections, this study decided to look at what happens with vitamin D and candida. And what they found was a bimodal response. And what that means is they found that people with candida tend to have a low level of vitamin D, and when you increased it, the ability to fight the candida increased. But if you kept increasing the vitamin D and went too high, your ability to fight the candida infection went down again. That this study shows a negative effect of going higher on vitamin D was interesting to me, because this other study on ALS also shows that if you go higher with vitamin D, you have a worse outcome. This last paper looks at the degree that candida integrates into the host with all the different strategies that it has in working around innate and adaptive immunity. There's considerable association with ALS and Lyme, and there have been a number of people that have been diagnosed with ALS and then later diagnosed with Lyme. So we're going to look at what are the complement evasion strategies of Lyme. So this paper shows that Lyme grabs onto the factor H, and that protects them from being killed. Okay, so what complement evasion of Borrelia on this particular study, this one's finding is it's inactivating the C3b. So it's taken the C3b out of circulation, and if you remember, the C3b does the amplification, and it also identifies the cell for killing. So if it's inactivated the C3b is not going to be able to attach to the Borrelia, and the amplification loop for innate immunity is also really turned down. Remember, when the complement parent protein splits, you get parts named A and B. So C3 splits to C3a and C3b. So this study on the split products for Lyme shows that there's increased C3a and C4a. With Lyme's complement evasion strategies, it is removing the C3b and the C4bp, so of course you have excess C3a and C4a. So now this next one, which says that takes 3 to tango, what this one is talking about is 3 components of Lyme. So you've got the host, you've got the actual Borrelia, and you've got the tick. And if you look closely at this one here, so what you've got is you've got all of these evasion strategies by the bacteria, the Borrelia itself, right? And you can see this first picture, these CRASP 1 to 5, they all grab the factor H proteins. And, this is a huge amount of factor H that this one here appears to be able to grab onto. And then you can see the C4 binding protein in this picture as well. And so it's also taken the C4B out of circulation. And if you remember, the C4B is what fed into the amplification loop. So you're not supposed to have a lot of C4B because it's supposed to feed the amplification loop. You're supposed to get tons of the C3B from a little bit of C4B. But this is pulling out the C4B. Okay, when you look at the actual Borrelia, you can see in the actual Borrelia that it's destroying the MAC complex, right? And the MAC complex is what actually goes on to actually kill the Borrelia. So it's got strategies to turn off the complex that actually kills the Borrelia. Okay, and then if you look further at this one here, you can see proteins from the tick also bind parts of complement. That binds onto the MBL, the mannose binding lithians, and it blocks them from doing their job. So this Borrelia has 3 different kinds of ways where it is interfering with complement. This study is showing that Lyme is also blocking the classical pathway through C1, and that again is in the initiation part of complement. Chlamydia pneumonia is more common in ALS patients, and what the body needs to fight it is something called properdine, and factor H and properdine have to be in balance for properdine to work properly. Herpes virus simplex 1 is very common in ALS, and it has an evasion strategy where it uses something called glycoprotein C to bind to C3b to prevent complement activation against it. So if we look at the big picture of complement, we see that interference with C1 slows down initiation through the classical pathway. Interference with C4BP slows down initiation through both the lithian and the alternative pathway. Interference with factor H slows down initiation through the alternative pathway. So we have huge interference with initiation. Then the actual amplification loop has further interference, turning it down by the capture of the C3b. Interference with MAC and factor H interfere with the attack, so far more initiation of innate immunity is required. And when we look at C4 of complement, which makes the C4BP, we see that there is a copy of HERV-K that lives on C4. So there's a huge overexpression of C4 because of the crippled initiation and amplification of complement. TDP43 is recognized as a problem with the misfolding of the proteins that are toxic to the motor neurons. This study says that TDP43 regulates HERV-K accumulation, but I think the overexpression of HERV-K is because of the crippled complement. TDP43 was first discovered to regulate HIV, and I think there's overexpression of TDP43 trying to deal with the excess HERV-K from crippled complement. Only there's so much HERV-K that TDP43 is unable to accomplish that goal. Aspergillus has no studies showing it linked to ALS. However, because it has factor H and C4b evasion strategies, I suspect that anybody that's living in a home with mold in it is probably going to have a greater problem with ALS, faster progression. There is this other study that showed a cluster of 3 people living in the same apartment that independently in an 18-month period got ALS. And if you're looking at an apartment building, what I suspect that they would have in common is potential mold exposure. The Leaky gut study found a microbiome shift from healthy to pathogenic microbes. There are several health consequences from this kind of shift. First, there's a reduction in butyrate. Butyrate is essential for the clonic EC cells. These cells make most of the body's serotonin, and it has a very important role in innate immunity. Serotonin is regulated by metabolites from the gut microbes, and there is an increase in mobility from increased serotonin. So you get the largest increase in serotonin from the most harmful microbes, which helps you to eliminate them. So there's a shift to microbes associated with disease, and the clonic EC cells lack sufficient butyrate to be healthy, which can impede their serotonin production and gut mobility, which can increase reabsorption of toxins in the gut. The second thing is the intestinal barrier of the gut is doubly compromised. You have reduced antibiotic protection because of that lack of the defensin alpha-5 that's right at the barrier of the gut, and the junctions are loose, so you have greater opportunity for microbes and large molecules to get past the lining of the gut. So the body's dealing with way more pathogens breaching the barrier and larger molecules getting through, so the body's secondary defenses have to work so much harder. And then a third very serious consequence of this kind of microbiome shift is reduced nutrition. Instead of having microbes that are giving you nutrients and making the nutrients you need, there are microbes that are competing for the nutrients that you have. ALS patients have an amino acid profile that has shifted to the point that it's been considered a biomarker for ALS. The amino acid shift suggests that the gut microbia have a compromised schidamate pathway. Now, we don't have this pathway in us, but it is in our gut. The essential amino acids that we get from the schidamate pathway are tryptophan, trypsin, and phenylalanine. And then methionine is also compromised with the pathway being compromised. Without sufficient nutrients in this pathway, we have a whole bunch of other things that are compromised in production. Serotonin, melatonin, melanin, epinephrine, dopamine, thyroid hormone, folate, coenzyme Q10, vitamin K, and vitamin E are all compromised for production. It is well understood that with ALS there is a serious problem with nutrient absorption. This research group is studying nutrient absorption after starvation, and they created a mouse model that does not properly absorb nutrients to study why nutrients are not absorbed when you just give a starving person food. And their mouse model microbiome has features in common with the shift in the microbiome found in the ALS leaky gut study. So looking at this overall, there is enormous evidence for fungal infection. The autopsy study that found the fungal infections, the galactin-3 is evidence, the bimodal nature of vitamin D response to candida is evidence, and this evidence shows up fairly early in the ALS mouse model with the galactin-3, starting at around 60 days and taking off. The leaky gut study also shows that prior to neurological symptoms, there is a microbiome shift in an ALS mouse model, and reduced defense in alpha-5 means reduced defense against fungal infection. Another point is that the microbiome shift is also deficient in the lactobacillus species that help to control fungal infections. There is also a serious amino acid deficiency related to the Schimmelmatt pathway, which may be related to the microbiome shift. This serious nutritional deficiency means that any system dependent on those amino acids is going to be crippled. Candida cripples all of the complement pathways, making people vulnerable to other infections. Okay, so my conclusions are that much of what is measured in ALS research is a result of fighting infections with nutritional deficiencies. These nutritional deficiencies will find genetic weaknesses, clouding and distracting away from actual cause. Something has to set the body off to have this microbiome shift, and that happens very early in the ALS mice models. At the very first sign of a neurological problem, people need to be tested for the microbiome and dysbiosis. They need to be tested for toxins and amino acids, and strategies to fix or help shift these things back need to be applied immediately. Indeed, maybe some of these people who have known genetic links had these strategies early, maybe their body wouldn't go out of balance. Working on this project changed my focus from what I had seen as crippled complement, with factor H and C4 having a very important role in the autoimmune development process. However, it also helped me to see that the body is set up to be crippled to fight back because of the serious nutritional deficiencies and the microbiome shift. Much about the disease seems to feed from these problems. With that, my thoughts go to long-term survivor Dr. Craig, who was diagnosed back in 1984. He got to the point that his breathing was severely compromised, and he went to hospice expecting to die. Only he didn't die and graduated 6 months later. Since then, he's gained back muscle function, and he believes that if he knew what he knows now about ALS, he never would have declined as far as he did before he started to heal. Dr. Craig worked with a nutritionist early in his fight against ALS. Another observation is that statistics say a certain percentage survive over X number of years, but my observation is that long-term survivors took committed action, and they also found the right kind of help. Serotonin is one of those downstream molecules that depends on the Schimmelmatt pathway, and it is a predictor for how long people survive with ALS. So I was doing a lot of research about how to fix serotonin, and this book has chapters on all of the things I was finding that are related to how you help your body make serotonin. Serotonin is related to depression, so it makes sense that a book fixing the root causes of depression is also fixing serotonin. The chapters include information on the gut-brain axis, cholesterol bacteria in the GI that affect dopamine and norepinephrine. Those are other downstream molecules that we notice depend on the Schimmelmatt pathway. It also has information about how stress affects this. Then there is micronutrient deficiencies and how that affects mitochondrial dysfunction, which is huge in ALS. Vitamin deficiencies, which also depend on the Schimmelmatt pathway. Mineral deficiencies, essential fats, amino acids, adrenal, reproductive, thyroid hormones, dietary peptides, how amino acids and other nutrients enhance synthesis and regulation of neurotransmitters. So it's all huge information to help understand how all this stuff fits together. There is evidence that viruses are playing a role in ALS, and this particular book is about how you improve your immunity against viruses. Kim Cherry is considered an ALS reversal, and this reversal did not come about on its own. Kim Cherry fought very hard against the disease with a wide range of strategies and a wide range of specialists. He talks about the terror he had at one time over having difficulty breathing when lying down and staying up all night because of it, and he has not had breathing issues for about 4 years now. He continues to manage his ALS. If he goes off his program, he does notice a problem coming back. And I've heard other long-term ALS survivors say the same thing. They must follow their program to remain healthy. Stephen Shackle was diagnosed with ALS in 1995 and has been in remission since 1997. He tells his story and shares what he has learned over the years of studying ALS. You cannot just give a person deficient in the nutrients and expect them to absorb it. He talks about what else various nutrients need to be properly absorbed. He also talks about how just trying to treat with amino acids doesn't work. Dr. Amy Yasko's information is amazing. She's an expert in DNA, RNA diagnostics, biochemistry, and molecular biology. The second link given is to a PDF from a presentation titled Microbes, Metals, and Methylation. And methylation is a problem in ALS. Page 63 of this document is the start of talking about viruses, and she points out they increase glutamate, which is a problem in ALS. The viruses cause heavy metal retention, and she backs that with enormous research. She has similar research showing bacteria causes retention of aluminum, which she again backs up with enormous research. These interfere with glutamate dehydrogenase, and mercury inhibits glutamine synthase. This presentation goes into great detail on how these mechanisms work. The aluminum inhibits the acetylcholinesterase, which is needed for the gut-brain axis and can cause neuroinflammation. She understands how limiting amino acids affect function. For example, with insufficient methionine, one of the amino acids from the Schmidt pathway, choline becomes insufficient, and you have a problem with S-adenylsulmethione, or SAM, and this just so happens to be one of the tested supplements that has some efficacy with ALS. She touches on mechanisms for many problems in ALS and knows where other problems show up. But just trying to treat one part of the system because of all of the mechanisms are connected just doesn't work. The Know Your Genetics link takes you to all of her resources, and she has her books to guide you through testing and how to fix problems found. She has a line of nucleotide products to help with DNA, RNA problems specific to your genetics. Given that there is a recognized RNA problem in ALS, her information has a lot of insight into mechanisms and where they can break down because of lacking nutrition. She has a whole series of videos that help to explain this stuff, although the teaching level is for a health practitioner. I did not find links to trained health practitioners. If you look at methionine alone, you can see many compounds listed that many people say helped their ALS. And this is why I'm saying that this nutritional deficiencies feed everything downstream, and her resources are designed to help people with this. I really love and appreciate what Dr. Tidone is doing for the ALS community. His program addresses nutritional deficiencies resulting in problems with the Krebs cycle in making ATP energy. ATP energy mostly comes from the mitochondria, and they are damaged in ALS. His daughter Deanna was diagnosed with ALS in 2006, and he has put himself and his family out there trying to help other pals. He has written a book, and he has managed to get studies done that show his program does help. However, it does not seem to work for all pals. If you read his book, one of the things that he did with his daughter was chelation therapy, and when I reviewed Dr. Yasko's stuff on how microbes mess up metals, I think that may have inadvertently been important. If you look at Shackle's stuff, he tells you outright that he's seen a lot of people do chelation therapy, and they think it helps them in the short term, but ultimately they do continue to decline. But I do question if it is the combination that's important. If a pals has heavy metals, it likely needs to be addressed. The smaller reference is not Dr. Tidone's, but I include it here to help explain why his program helps. By 65 days in the mouse model, there is evidence of a switch to lipid metabolism. They use dichloroacetate to help force the continuation into the Krebs cycle, which delays denervation. I believe this switch to lipid metabolism is huge in what is causing oxidative stress in ALS. Our bodies are designed to get ATP energy from the Krebs cycle, and in times of need, supplement with lipid metabolism and then take a break and heal. With ALS, lipid metabolism is on full time because of the damaged mitochondria, and it is doing enormous damage. I think coconut oil helps because it has medium-chain triglycerides that the body is able to use for energy and bypass the step with the most oxidative stress damage. Deanna also applied coconut oil to her skin daily, so I think there's 2 levels of energy support in the Deanna protocol. A very important note here, Deanna has been recently found to have Lyme disease and is being treated for Lyme disease, and I hear she's getting stronger. And a final point, a number of pals say that apple cider vinegar helps them, and this dichloryl acetate is a salt of vinegar. Dr. Nicolson runs his Molecular Medicine Institute and has a number of published papers on lipid replacement therapy for healing mitochondrial cell membranes. All of his papers can be accessed on his site. The 4 lipids to replace cellular membranes are the P-choline, the P-inositol, the P-ethylamine, and the P-styrene. Those blends are available from the NT Factor site. For me, the one that stands out is the P-serine because Kim Cherry did not take a full lipid replacement, but he did take this one twice per day, and he is considered an ALS reversal. So I know that complement is incredibly compromised in ALS. The title of the paper I've included on the slide is not Dr. Nicholson's. However, it is about what I think is an important mechanism to get rid of diseased cells. To me, this is an area that we need specific ALS research, as I've also seen testimony from my pals about being sick for a couple days from trying an NT Factors product. So there is research that shows the destruction of cell membranes around mitochondria in the livers of ALS patients, and this is a problem that does need to be addressed. This paper seemingly has little to do with ALS, but what this paper is showing is an extremely important mechanism. Cystic fibrosis is recognized as a chloride channel blocked disease in the lungs. Thiocyanate uses the same channel, and so the disease of the lungs is a model of disease when there is no thiocyanate. And what you see in cystic fibrosis is enormous scarring of the lung tissue and huge mucus buildup. So what this paper is saying is that thiocyanate is a necessary molecule for the neutrophils in their chemical warfare against pathogenic microbes. And what the thiocyanate does is it rapidly neutralizes the chemicals that kill the invader, and it limits the collateral damage to host cells. I don't know if the neutrophils can actually survive the attack and attack more pathogens with adequate neutralization of the chemicals they use from this lactoperoxidase and myeloperoxidase oxidation reactions, but most of the mucus in cystic fibrosis is from the death of host cells because of the inability to neutralize and limit this reaction in the neutrophils. So in ALS, just by virtue of a leaky gut, secondary defenses will be fighting far more pathogens, and it seems to me at some point there will be a deficiency in thiocyanate. And so at any site in the body where neutrophils lack thiocyanate, there will be more damage to host cells, and this will speed up disease progression, not to mention increase the mucus secretions. I've not found any papers that measure thiocyanate levels in ALS. The cystic fibrosis study has a discussion about optimal levels of thiocyanate. Thiocyanate is a micronutrient with dangerous toxicity with too much, and the toxicity level is easily reached. Desperate people tend to think more is better, so there's a lot of literature on the harm, but I don't think the information is balanced with actually looking at what neutrophils do with thiocyanate. There are many papers showing benefits to thiocyanate. The link is for where you can purchase the book with this title, and you can find a summary at Mercola.com. Some words that stand out in this article: hypercoagulation, biliary sludge. I think this is where the genetic links to genes involved in the ciliodome come in. Cilia help with mechanical movement in the body, and if you have a coagulation happening, say in the bile ducts or the lymphatic fluids, problems from poor movement of wastes and nutrients in the body is going to be compounded with impaired cilia. From the article, abnormalities of the hippobiliary system may involve biliary stasis, where infectious materials or biotoxins reside in the liver, biliary tree and gallbladder as viscous suspensions in biliary sludge. I really believe that ALS has a mechanical movement of nutrients and waste problem along with nutritional deficiencies. For example, the bile is supposed to feed manganese to the lactobacillus, and they are manganese oxidizing species, but for some reason the lactobacillus are not there, and they are a major defense against candida. Manganese getting backed into the liver is toxic to the liver, and this may be why the manganese builds up the spine, to release it from the liver. But coagulation and viscosity problems have to precede this for it to make any sense. Healing ALS has a few goals. One is to make a documentary about the people who have healed from ALS or who have managed to survive long term and tell the story about what they did. Another goal is to change the stigma that nothing can be done when often nothing is done. There is some very practical starting tips and testimony of people who found the right help and made the right choices. I've talked to a couple of these people, and they are managing their ALS. They follow their program to stay healthy, but they do notice a problem if they cut back on their program. Derek Sweenard is an 18-year survivor of ALS, and he is documented on healing ALS. He credits the help from free-date homeopathics as the reason he survived. Dr. Alan MacDonald is an expert on Lyme disease. The 3 links are to videos that give a comprehensive presentation about Lyme disease. One of the things that really got my attention in the discussion is the methodology on how Lyme testing was developed. With this kind of methodology, testing for Lyme can confirm some people have Lyme, but it can miss if there's any genetic diversity in the Lyme species, so a negative test does not rule out Lyme. The bottom link is a press release about results of brain autopsies of patients who had serious neurological diseases, and they show tick-borne disease. When people are re-diagnosed with Lyme disease, there are people with ALS that will say, well, that person really didn't have ALS. And my point here is that Lyme goes undiagnosed and can give ALS symptoms, and current testing does not rule it out. So to finish this video, I returned to Hazel. I only knew Hazel online, but her drive to find what was killing her was infectious, and her excitement at gaining back movement and seeing it happen was joyful and powerful and hopeful. In 2014, Hazel, like myself, really didn't know what ALS was, and what seemed like fun to participate in was going to be front and center and dominating all life moving forward. Hazel's progression was fast. Diagnosed in September of 2015 and completely paralyzed of her limbs by March of 2016. Hazel found a doctor who started to treat her for a Lyme co-infection with a drug called Rayomet, which is an anti-malaria drug, and then a series of many other antibiotics for Lyme. She ran into problems and was looking for help that no one knew how to give about 2 months into her protocol. She was feeling much better the last week of June, and so very excited to be re-diagnosed with Lyme on June 30th. The next day, she passed away from a stroke.